This is the third presentation in the series Getting to Know Your Genome. In this presentation we will look at what genetic information is, how genetic information is stored, and how cells use the genetic information. As we have seen, genetic information is passed from the mother and the father to the offspring. This happens when the egg is fertilized by the sperm. We call this heredity and say that a child has inherited genetic information from its parents. A person has many characteristics that are similar to its biological parents. Genetics is the science that studies how genetic information shapes these characteristics. We have also seen that the genetic information we inherit from our parents is stored in pairs of chromosomes in a genome. The genome is in the nucleus of each of our cells. A chromosome consists of a single molecule of DNA, deoxyribonucleic acid. The DNA is folded around protein molecules called histones. There are 46 molecules of DNA inside each cell nucleus. It is important to realize that the genome stays inside the nucleus of the cell at all times. The nucleus makes a copy of the genetic information which it sends out to the parts of the cell that make protein. As cells are continuously making proteins, the nucleus is a very busy place. Genetic information is a set of coded instructions for building proteins. Think of it like a cooking recipe or a knitting pattern. The cell uses this information as a template for building protein molecules. To understand how the genetic code works, we need to know about the structure of a DNA molecule. DNA has two interconnected helices held together by weak chemical bonds. It is called a double helix. The outside of the molecule is called the backbone. It is made up of many sugar phosphate groups held together by strong chemical bonds. We call the backbone of the DNA molecule strands. The sugar S is deoxyribose. Attached to the sugar is a phosphate group P and also a base. The sugar phosphate base together make up a unit that we call a nucleotide. A DNA molecule consists of many thousands of nucleotides joined together. There are only 4 possible bases in a nucleotide: adenine A, thymine T, cytosine C, and guanine G. So there are 4 different kinds of nucleotide in DNA. The double helix of the DNA molecule forms when nucleotide bases on opposite strands form weak chemical attractions to each other. There are strict pairing rules for these bases. Adenine only pairs with thymine, and cytosine only pairs with guanine. So A pairs with T and C pairs with G. We call the nucleotides that pair together nucleotide base pairs. DNA is made of pairs of nucleotides held together by weak chemical attractions. There are 6 billion pairs of nucleotides in the genome in the nuclei of your cells. But there are only 3 billion nucleotide pairs in your sperms or your eggs. Let us look at the sequence of bases along one strand of the DNA molecule. A, G, C, T, C, G. We can see that they can be written as a string of letters. This is called the genetic code. To interpret the code, we have to group the letters into sets of 3. These are the instructions that the cell uses to make a protein. Proteins are made of long chains of amino acids held together by chemical bonds. The DNA code tells the cell which amino acids to join together in which order. A set of 3 nucleotide bases in the DNA codes for 1 amino acid. Proteins can contain hundreds of amino acids, so many nucleotide bases code for each protein. A gene is all of the nucleotide bases that code for 1 protein. There are about 28,000 genes in the human genome. Even so, genes make up only about 1% of the complete human genome. The remaining 99% of the genome does not code for protein. It is distributed between the protein coding gene sequences on every chromosome. The function of non-coding DNA is not fully understood, but it seems to help the nucleus to process the information in the genetic code. People usually have identical proteins, so it's not surprising that the nucleotide bases that code for those proteins are the same in all of us. About 99.9% of the human genome is the same in all people. Proteins are important because they form a major part of the structure of a cell. Also, as enzymes, proteins control the very many chemical reactions that occur inside cells. Proteins are important because they work together to produce the complex characteristics that humans have, characteristics like height, weight, intelligence, and so on. Proteins are also active in the development of certain medical conditions like autism, dementia, and schizophrenia. The diagram shows some of the interactions between proteins in a person with schizophrenia. Human genetics can only study differences between people. So this course will concentrate on the 0.1% of the genome that shows differences between us. We call these differences genetic variation. Mostly, the genetic variation is between nucleotide bases that do not code for proteins. Even so, that is still 6 million nucleotide base pairs. These are distributed across the 46 chromosomes in the nuclei of all of your cells. So, what does your genome do? The genome contains information that the cell uses to make proteins. DNA contains nucleotide bases that act as a code for the production of proteins. A sequence of 3 bases along one strand of DNA specifies one amino acid in a protein. Proteins are made up of hundreds of amino acids, and therefore thousands of DNA bases are used to specify the structure of a protein. Proteins are important in controlling the way that cells and organisms grow and develop. A gene is a sequence of nucleotide bases that code for a protein. There are 28,000 genes in the human genome. Most of the genome does not code for proteins. Genetics studies the difference in nucleotide bases between people. This is called genetic variation.